one and all present here. I am Bakha Goyal from final year CBPE, student coordinator of Transgene Club. I, on behalf of the management, staff and student fraternity, welcome you all to this wonderful gathering. Now, I request Dr. Pratibha K.S. Ma'am, Head Department of Biotechnology, to welcome the gathering.
Good afternoon, dear students.
Good afternoon, ma'am.
Good afternoon, dear students.
Good afternoon, ma'am.
Thank you. Kindly respond. Today we are here for interactive workshop on bioinformatics and research methodology, which is based on your paper 8. This workshop organized by IQAC, by the Department of Biotechnology. Today we have with us eminent personality as our chief guest of this interactive workshop, Dr. Pavan J. Kundur, Assistant Professor, Department of Biotechnology, GM, Institute of Technology, Davangere. On behalf of KLE Society's S. Nijinappa College Principal, Management and Student Fraternity, Faculty, and also on my personal behalf, we express sincere thanks for accepting our invitations and welcome you to this gathering. And I also welcome one of my colleagues, Mr. Rajiv R. Potadar, for this interactive workshop. Last but not the least, I welcome all our dearest young and energetic students for today's interactive workshop on bioinformatics and research methodology. Thank you very much.
Thank you, ma'am. I now request Mr. Rajiv Potedar sir, Assistant Professor, Department of Biotechnology, to introduce today's eminent resource person, Dr. Pavan J. Kundur sir.
Very good afternoon to everyone.
Good afternoon, sir.
Respected Dr. Pavan J. Kundur sir and respected Dr. Pratibha K.S. HOD, Department of Biotechnology, and my dear students. I'll take this privilege to introduce the chief guest of today's the interactive workshop. So Dr. Pavan J. Kundur, he has completed his the post-graduation from Queen's University and Bangalore University. So he has secured 2 PGs in zoology and biotechnology respectively, and he has completed his PhD from VTO, Belgavi, Plant Biotechnology, on thesis Molecular Genetics of Root Growth, Ion Absorption and Transport in Rice under Aerobic Condition. So he has around 12 years of teaching experience and 7 years of research experience, and he has contributed 3 different research projects funded by Karnataka State Council for Science and Technology, and he has recognized as a Board of Studies and Board of Examiners for KLE's RLS College Belgavi and KLE's PC Jobin Science College, and he has 13 research publications to his credit, which includes national and international publications, and he has 2 patents filed to his credit. So in the field of mineral water quality testing system and Indian enhanced storage method of gravitational energy to manage renewable resources. And he has published 5 different book chapters which are published in the ISBN and ISSN numbers. And he has participated and presented more than 10 the conferences and seminars, and he has organized so many events as the coordinator and convener of the different events, symposia, workshop, FDP, etc. And he has guided the students for the postgraduate and undergraduate research funded by Karnataka State Council for Science and Technology, and he has received a Best Oral Presenter Award for oral presentation on implementation of artificial intelligence for sustainable agriculture in international conference organized by the KLESNC College. So I welcome sir for this interactive workshop. And to add up a few the recognitions, so he has developed two important products, biotechnology product. One is areca pickle, and another one more is vetiver root-based water filter. So he has designed these products as a biotechnologist. Thank you, sir.
Thank you, sir. I now also welcome, on behalf of the management, staff, and student fraternity, Dr. Pratibha K.S. Ma'am, Head Department of Biotechnology. We welcome you, ma'am. Now, I request Dr. Pratibha K.S. Ma'am, Head Department of Biotechnology, to present a token of gratitude to sir. Thank you, ma'am. Thank you, sir. The application of tools and computation and analysis to capture and interpretation of biological data is bioinformatics. So now, let us all grab some more information about what actually bioinformatics is. So now I welcome sir upon the stage.
I hope
Respected HOD madam and Rajesh sir, thank you, and also its application. So we'll get into this, right? So in 45 minutes of session, it is very difficult for us to interact. So with this biggest constraint, so we will now I will make all of you need to understand so what is the basics of this, and later on if time provided, so we will discuss with the staff of BT, and we will further discuss about the concepts in bioinformatics. So have you heard about bioinformatics earlier, students?
Yes.
Okay, how many of you sitting here are interested to do computer science, and you didn't get the chance to do it, and you are here, sitting here? So none of you, right? Still any chances? So if you want to become a software engineer or a computer science engineer, if you get chance, so he's saying yes, right? So if you want to become, so you can become a bioinformatician. So it is a emerging and trending world, so which is happening in 2023, and also forecast of 2030s also. So let us understand what is bioinformatics is all about. So bioinformatics is all about, so if you split the word into two, so it is all about bio plus informatics. So what is bio? Bio? Living life. Yes. Right. Any other answers there?
Life.
Life. Very good. So informatics. Information technology. Very good. So it is a combination of bio as well as the information technology. So bio means life. So already you know that information, and life is DNA, right? And life is DNA. So why life is DNA here?
Every living cell has DNA.
Okay, so since for all our metabolic activity and reproduction and other activities, so we need DNA as a genetic material which is inherited from our parents, right? And it is also required for growth and metabolism and reproduction. So without DNA, so you cannot
Okay, expect life on Earth. So we all okay. So all of you studied the central dogma, right? So what will happen in transcription? Transcription. DNA. So DNA convert to mRNA. So what is mRNA? It's a messenger RNA, right? So what is translation? mRNA to proteins, right? So proteins are the one. So which what they will do? So they are functional in nature, and whatever the activities you want to do, so they will help you, right? So it is all depends upon your behavior, your physical appearance. We call phenotyping. Genotyping, you know the difference between phenotyping and the genotyping, right? So whatever the gene makeup is there, so that is what you will look like your parents or grandparents. So that is how it goes, right? So that is the combination of both the transcription and translation called central dogma, right? So informatics is all about data collection. So what type of data collection? Okay, so depends upon the discipline. So in this campus you have arts and commerce and science, right? Right? Yes or no?
Yes, sir.
So in that case, the data collection is differs from each branches. So in biotechnology or in life sciences, it differs a lot, right? So what can you give one example? Okay, for biological data, students here.
Gene library.
Okay, forget about biological data. Any data,
Gene library.
any data you come across in your day-to-day life. Your attendance, your classroom attendance itself is a data, right? Yes or no? Yes, so that data, okay, so who are all having shortage of attendance and who are all having right 10% attendance, so that is where you are going to statistically analyze at the last. Okay, so here you are not going to do. And data management, so that is how. So collection is not your task is done. So very important thing is you need to manage it properly. Okay, and we will have some information, okay, in later place. And information, so what type of information it is, so whether it is useful for your research or whether it is useful for your innovations or whether it is going to helpful for the mankind. So that is what we call it as information. And next one is information exchange. See here you can see, okay, one example I want to give here. See the development of COVID vaccine. So it is not a simple okay, so they have they have extracted okay, the proteins, so which are coding for antigens of that particular virus, and they have okay, developed the COVID vaccine, the Covishield and Covaxin, right? So what they have done, they have collected the data from okay, the whole world, right? The first time what they have done in 2020, the Chinese okay, the Department of Health, so they have okay, so collected the genomic information of that COVID-19. So they have published into the database, right? So by taking that database as a reference, they have developed the product. So which is what we are talking about, the vaccine development. So behind that, so lot of work has been taken place. Okay, so that we are not aware. Okay, so that is what we deals with the informatics, right? And exchange of information, that is what I mentioned here. So the information was exchanged so that it is very easy for scientists to achieve the product. Okay, so the end product is vaccine. So now we will get into the okay, the core of now you got so what is all about the very short trailer of what is bioinformatics, right? So bioinformatics is a branch of biological science that has emerged from the combination of both biology and information technology. So it comprises of chemistry, biochemistry, biology, statistics, mathematics, engineering, and also computer science. Okay, so different. It is a multidisciplinary department, right? And here the bioinformatics mainly involved in analyzing biological data and developing a new software using biological tools. So different tools are there. So for example, okay, so in your Instagram, Facebook, the filters are there, right? In your cameras also, filters are there. Okay, to enhance the beauty of an individual, right? So here also to enhance the beauty of okay, the biological data what you are collected. So that I will call it as raw data. Okay, how you are going to interpret? So that is what these tools will do. Okay, so if you want to really okay get into the the software development for bioinformatics. So you need to acquire some knowledge of programming. You need to learn SQL as well as Python programming language. Okay, SQL as well as Python programming language you need to learn, so that you will become a bioinformatician. At the last of this presentation, I will tell you where and all opportunities are there for the bioinformatician. So, history of bioinformatics, I will just brief through this. See, in 1965, Margaret established the first database of protein sequences, published in entitled Atlas of Protein Sequence and Structure. See, students here, so they have published, but there is no such the information exchange method. For example, what is your good name? Rohit. So Rohit is working at India, and what is your name? Divya. So both are working on the same organism. Okay, so I will give one example, pre-bacillus species or E. coli. So E. coli is better to remember. So you are both are working on the same organism, and you have published the same output or data right into the biological data system. So if you know that someone has already done that work, okay, so what you would have done? You would have just avoided it, right? So that is what this informatics and the data will help you. Clear? Right? So what it will help? So the repetition of the same work which is done by the others. Okay, so that is very much important, because the lot of efforts and the resources you have already spent there. So what you need to do? You need to look for the whether this product is available, this research work is already which is performed or not. So that information where you will get? You will get into this database system. Okay. So and during 1977, the DNA sequences began to accumulate slowly into the literature. So what they have started, whatever the information they used to get, so they used to publish and print it into the books, okay, or papers. So that was it was not published into the data system or warehouse. So what we call. So and whatever the work it has been carried out, so none of them they used to know. So for example, if you want to perform research, okay, on some domain, so you need to go to the China and you need to get that information. Okay, you need to copy that and you need to come back to India and you need to do that. You need to continue that work. So, but now into your fingertips, so you are going to get all the data which is been already published somewhere. Thus, number of uncharacterized proteins are begin to increase. So, what is the meaning of uncharacterized proteins? See, number of proteins are there. For example, so if you want to develop, okay, so now next for final year or final semester, you are into final semester now. Okay, so you will be having project works, right, or internship. So in there, what you will do, you need to go to some Western Ghats and you need to find out some medicinal plants and you need to isolate some compounds which cures cancer. One example I am just quoting here. So at that time, so you need to specify. So what is that protein is? So without that you cannot so give it to the patients. So that we will talk in precise medicine. So it is one of the best applications of bioinformatics, right? So without this knowledge, so you cannot achieve anything, students there. So in 1980, there was enough DNA sequences justified established, and they have established the gene bank. So it is called as gene bank. So if you type NCBI, so into the Google, so you will get this information. And so they found NCBI, so USA, National Center for Biotechnology Information. So what are all the data you have collected? For example, it may be a DNA structure or a protein, the sequence, a DNA sequence, ATGC. So in protein, what will be the sequence? Amino acids, the 20 amino acids. So that is the sequence there, right? And the next one is NCBI serves as primary data bank provider for the information. Since it is the first and foremost database, so which is established at USA. So now, till today, so it has been started. So it has having the depository of 65 lakh crore nucleotide information. Means 6 trillion in database information. All over the world, the scientists are deposited into the database, right? So the next, after that, so they have established European Molecular Biology Laboratory (EMBL). So this is very important, students. Abbreviation. Okay, so if you miss that abbreviation, so you are not going to fetch the very good marks. So abbreviations are very important here. Okay, so where it is established at European Bioinformatics Institute. Okay, so that is abbreviated as EBI. Which year? Which year? 1980. So in 86, DNA data bank was established by Jamonnet, Japan. Okay, so in Japan also they have started. See, at the 3 different locations. First one is USA, second one is Europe, and next one is Japan. Okay, in Asian continent, Japan, right? So remember, so in US, American continent, and Europe, European, and next one is Japan. So earlier what they used to do, for example, I will categorize these 3 rows as, okay, so USA, Japan as well as Europe. So whatever work you used to do, you used to publish in EMBL only. So whatever work you used to do, so you used to deposit in DDBJ. So no information was shared. You are understanding? So no information was shared. And the people who are sitting in the research labs of USA, they used to perform some work and they used to publish in NCBI only. Okay, so that information they were not knowing. And but fortunately, they have made a collaboration. So that is what we call International Nucleotide Sequence Database Collaboration, abbreviated as INSDC. So with this INSDC, so today, so if you wish to, okay, get the information of BRCA1, so Cry1AC. So what is Cry1AC? Cry1AC.
BT protein.
What is Cry1AC?
It's a protein.
Very good, gene. So where it is used? BT cotton. Okay. So if you want to get the sequence of Cry1AC, so if you search in EMBL, if you search in DDBJ, if you search in NCBI, you will get the same result because of the collaboration. Okay, so because of International Nucleotide Sequence Data Collaboration. So in 2023, so what is going to happen? Okay, so the bioinformaticians predict we'll see a lot more research in spatial transcriptomes and also immunotherapies for cancer. Okay, so immunotherapy, so what it will do? It is a drug, so very costly drug. So if you are having any solution, you do the same one. So if you reduce the price of that particular injection, so you will be awarded okay, so with the honor. Okay, so because one injection cost is around 2 lakh rupees for immunotherapy to survive cancer patients. So if any further research into the immunotherapies for cancer, so definitely it is going to help the poor patients, those who are suffering from different types of cancer. So how is bioinformatics used in the biotechnology industry? Studying and managing biological data. Okay, studying and managing biological data. So I have so given some information about biological data. In detail we will discuss. So next one, developing new drugs and therapies. How? Okay, so that also we are going to study. Okay, so how we are going to develop new drugs and therapies for different diseases. And improving the agriculture productivity. Okay, how you are going to, by use of bioinformatics, how you are going to change the lives of farmers. Right? Right? So this year, 70% of the farmers are awaiting for rain because of there are no monsoons enter. Okay, so they are predicting. So this year may be the sign of drought into system. So how you are going to develop crops with drought resistance? Is there any probabilities are there? Is there any chances are there? So for that, what you need to do? You need to look for the drought resistant genes. So in somewhere, okay, so if you take any weed without watering, so it will grow. How it will grow? Think, right? Maybe some genes are there, so which are helping it to grow it in very less water management. So if you transfer that system into your main field crops, for example, rice, paddy, rice, both are same, paddy or maize or any cotton plant, so what not? Okay, so if you transfer that, so what is going to happen? So that is what you need to think being a young biotechnologist. So computational tools to analyze, manage vast amount of data generated in the biotechnology research. Okay, so in biotechnology, for example, if you are doing molecular analysis, okay, so or molecular markers if you use, so you know what are molecular markers, students? Yes. So what these molecular markers will do? So So they will help to trace where exactly the gene is present. Okay, so these molecular markers help you to identify where exactly your interested gene is present. Clear? Okay. So for that, different types of molecular markers are there. So we have studied SNPs. Okay, single nucleotide polymorphism. So why you are different from your friend? Why you are not looking like your friend? Tell me. So it is all because of single nucleotide difference. Okay, SNPs, right? So because of one nucleotide difference, so you are not looking like your friend. Okay, so that is one thing you need to remember, students. See that bioinformatics can help scientists to develop new ways to diagnose and treat the disease. See, now we are scientists are talking about precision medicine. So now in a house, okay, so for example in COVID duration, almost all of them are infected with the COVID-19 virus, and the treatment it differs from individual. So why it is so? So it is all because of their genetic makeup. So already I mentioned, okay, so SNPs, SNPs are different. So now what the doctors will do, if you are suffering from COVID-19, so they will give the same treatment as others get. So now it is not going to happen. Since the human genome project is completed, so you are going to get tailor-made or precision medicine for you. So only exclusively for you. Okay, so depending upon your genomic analysis. Understood, students? Okay, so it is become so much precise so each one individuals who are living on the earth, they are going to get the different medications depending upon their genetic makeup. Clear? Okay, so that is going to happen shortly, but only one drawback is there. Okay, so if they are having your genetic information, so chances of misuse. So that is only one threat. So why they are not into the precision? What is the threat here? You know, organ donation scam. So in lot of movies they have exposed it, right? If any high profile politician or the rich people, if they need O positive and with no cholesterol and all these biochemical tests they have passed, right? You are understanding what I am saying? So what they will do? So they will force him to give the kidney or liver or any other organs. So but that is going to happen? No, never. Who will give? If I ask you give your kidney, you will give? No. So it is going to turn into a mafia. Okay, so if they know all the information, if they know all your profile, so definitely they are going to make you threat, or they will create a screenplay that so it is been an accident, right? So in one movie, so in South Indian movie Joseph, so they have shown so how this mafia work, right? So produce more efficient enzymes and vaccines and designs for better crops. Okay, so this is what we will discuss. So what are the objectives of bioinformatics? Development of new algorithms and statistics for assessing relationship among a large set of biological data. Okay. So what is this algorithm? Okay, it is a set of problem solving coding. Okay, in computer science language, it is a set of problem solving coding. Some coding you will do for that. Okay, your problems will be solved. Okay, so in research methodology we will talk more on this. Okay, so what is the problem you are suffering, and now how the startups in Bangalore, so how they are going to solve your needs. So you wish, okay, so I am not feeling well today, so I wish, okay, the food come to my doorsteps. So now Zomato is there and Swiggy is there. Clear? So I cannot go in search of auto, taxi, right? So now if you just pin your location, so everything the service will. So that is how the necessity is the mother of all the inventions. So and the application of these tools, the analysis and interpretation of the various biological data. So how best you can use these available data. So that is what you are going to understand in the objectives. It is one of the objectives of bioinformatics, and development of database for efficient storage, access, and management of large body of various biological information. See earlier, so who are having 4 GB memory card in cell phone, so you used to you were having 4 GB data, so I have only 512 MB. Okay, so and now it has changed, right? A 16 GB and now okay, so 256 and in some 1 TB also has come in Samsung. Okay, some of the cell phones, right? So why? Because the quality of okay the data it is increased. Earlier photos, so only 512 KB size. So now 12 MB high resolution 1080p photos. What is the size? 12 MB, right? So if you record video in 4K, so you okay, so now you cannot imagine, right? So in 1 minute of video, so it will consume lot of hundreds of MBs. Okay, so this is what the advancement. So that is what you need to stress here. So about the efficient storage and access management of okay for various biological information. How this is being done? So we will okay. So you are going to understand now. So the component of bioinformatics data, database and data mining tools. Okay, so data. So what you are going to create or generate into the laboratory, and the database where you are going to deposit it, because you cannot just print it and you can keep it, right? So the chances of misusing will be there of the highly confidential data. So especially in case of whenever you are looking for development of a new drug, students, right? So next one is data mining. So what is data mining? Searching for so your interested field or arena information. So that you need to dig it out. So for that the terminology is data mining. So data, okay, so from where and all you will collect the data. Okay, so one example is geographical, so location. So that now loggers, so they will go to some Masal Dosa, Vidyarthi Bhavan, Chamarajpet, right? They will go to MTR and they will make. So this is also kind of a data. So what is the price? So where it is available. So it is also you need to consider it is a data, right? Usually if you go Sari, okay, so our side and down here Beldango side is very famous. So where how it is being manufactured and how it is cooked. So that information you are going to get in geographical data. And what is the rainfall in down here now? Okay, so the percentage. What is the centimeters number? Right. So this is all about related to some geographical data where it is stress more upon the location of the place. Right. So next one is cultural. So how many the performers are there in this classroom? So that data your cultural coordinator will be having. So that is what it comes with cultural and the scientific. So that what are all information generated into the lab and financial. Okay, so what is my income and how much I have spent. So this is nothing to do with okay students of this generation, but your parents will take care. And isn't it? Okay, the financial burden, everything will be taken care from your parents. So that should not happen, and you need to take care of your okay the financial management. So it will be easy for your future life. And next one is statistical and meteorological means the how much rainfall. So already I mentioned it. And the natural. So how many trees were there? How many lost today? So in Bangalore, over 1 lakh trees are chopped down because of metro construction. So it is a data. Okay, so that you need to take and you can okay, so try to improve or replant. Okay, so wherever afforestation is being taken place. Transportation. How many okay women or girls in this class they have utilized free bus? Okay, so it is related to data related to the transport. I am just giving information. Or how many buses are transporting from Bangalore to Tirupati, Bangalore to Chennai? How many passengers they have traveled in it? Okay, so with the concession. So all these okay information is data. So everything is data. So now data you can okay in the form of text, observations, figures, image, numbers, graphs or symbols. So anything may be a data. For example, the data might include prices, weights, address, ages, names, temperature, dates or distances. So everything is considered as data. Okay, data is a raw form of knowledge and it is on its own, so does not carry any significance or purpose. So until unless you draw a conclusion, or you plot a graph or you come to a conclusion, so this is what happened. So it is not going to give you any information. So till that it is what? It is a raw form. The raw form or raw data is one, so which is not going to give any full stop. Okay, it is still something continuation is there. Biological data. So now I will stress okay, more specifically. So this is again a 10 mark question or a 5 mark question depending upon the requirement. So what is biological data? Okay, the data so which is generated so from the laboratory. So one example you can take, not a problem. Okay, so from the laboratory or any scientific results. Okay, so what are those raw DNA sequences? So you all know, students, okay, the sequencing. Okay, so Maxwell Gilbert and Sanger method of sequencing you have studied, right? So which is most okay, so widely used method of sequencing adopted? Sanger method of, it is also called as chain termination method of sequencing. So all of you know why is there? Okay, so it is very difficult to handle you after okay, so lunch. Okay, so please connect it with me. Okay, for another 30 minutes, so that you are not going to okay sleep. So please ask if you want to. Okay, if you are having any queries, so please feel free to ask. So next one is raw DNA sequences. So what are the sequences generated from your laboratory? So that is what we call it as raw DNA sequences. What is the use of getting the raw DNA sequences? What you will do with that? Is the end? Okay, so very good. So by taking this data, see, it is not that you have done, okay, so you have submitted some sequence of some organism or plant, okay, or yeast, whatever, unexplored, okay, organism. It is not that you need to conclude. So some any other who are sitting in this, so they can, okay, so continue the piece of work. So whatever others have done. So that is the main USP of bioinformatics. Okay, so whoever sitting, okay, so any part of the world, so they can utilize the data free of cost. Only the data which are patented that you cannot steal. So what you need to do, you need to write a letter, sir, I want to do this, and if he convinced the original inventor, so then okay, so you have no objection, you can utilize the data and you can start interpreting. Okay, and next genomic sequence tags. So here in the genomic sequence tags, the whole genome. Okay, so already the sequences are being published. So into that, so which are all functional one? For example, students, how many of you know the mice? So which is used as experimental animal. So into our okay, the in biomedical experiments as well as some of the biotechnological tools. So it is sharing 80% of the genetic information, right? So when you compare, so these two we are sharing 80% of genetic information. For that reason, it is one of the animal, so which is called as model organism or model animal for the okay any clinical trials which you are going to do. Okay, as a drug inventor, new drug inventor, right? So by doing that, so you are going to conclude. So what is the effect on the water filtration system? For example, kidney, right? So such type of okay, sir, conclusions you can draw. Okay. So and the cDNA, complementary DNA sequences. So are the one so which are useful in okay, so conversion of a single stranded to double stranded one. So that okay, so in sequencing we will study. And expressed sequence tags are present in the sequence randomly. They are present. Okay, so you know the length of human genome, students, 3.2 billion base pairs, right? So out of 3.2 billion base pairs, how many are present? How many ESTs, expressed sequence tags, are present? So that you are going to study. So what is that? It is a marker. ESTs are a marker. So like SNPs, EST is also marker, and also microsatellites also marker. Okay, so RFPD, RFLP, so they are also markers. Okay, so differences are there, PCR based and non-PCR based marker systems. And okay, organellar, okay, DNA sequences, the whole organism. For example, okay, so now we are only focusing on genetic metal which is present in the nucleus. So now you can do it from chloroplast and mitochondria also. Cell organelles, okay, that genomic sequence, sorry, the sequences also you can store. And RNA sequences, you are aware, the virus information, virus genetic information, it is stored in the form of RNA sequences. Next one is protein sequences. Okay, the protein sequences are one already in the beginning. I have just given the example. So whenever you have invented or developed a new drug, so what is the protein which is present? So how it is being given to the patients? So for that students, we call molecular docking. Okay, so docking system. So what you will do, you will design a compound and you will get the information. So by doing some simulations. So do you understand what is simulation? In simulation, Andre? So you are mimicking the whole event, okay, in a miniature form, or the same set of models have been kept there. Okay, it is just like you do the reels, right? For already the famous movie reels or dialogues, you will do it, right? The similar kind of what is the reaction, so that you need to understand, correct? So that is what we say here. Okay, so what is that? Protein, okay, sequences. And okay, so since they are a macromolecule in nature and the larger in okay size, so the protein structures have been the 3D modeling, and also the structures also you can visualize it. Where PDB, okay, protein data bank, and also Rasmol software is there. So by using Rasmol software, so you can visualize the 3-dimensional structure of any protein. Clear? And next one is metabolic pathways. So in order to understand the metabolic okay disorders, or if any, so in order to come across that metabolic pathways, so are very much helpful. Gel pictures, okay, in after performing gel electrophoresis, so you are going to draw number of gel pictures images, so that also you are going to store here. So what is the use? So later you need to score in a binomial nomenclature, that is 1 if band is present, 1 if band is absent, 0. Clear? So for that what we call, so gel pictures and literatures, obviously. So without literature survey you cannot achieve anything. So gene bank, right? So it is a comprehensive public database that contains over 6.25 trillion base pairs from over 1.6 billion nucleotide sequences for okay 4,50,000 formally described species. Okay, from how many species? See, the scientists across the globe, they have already done a majority of work. Okay, so you need to find out what is not done. Okay, see, look at the number 6.25 trillion. Already I just if you convert it into Indian rupees, 60,000 crore base pairs. 60,000 crore base pairs. I don't know how many books are required to print this. Okay, so how many books are required to, maybe it will fill almost the Central Library of Bangalore University. Okay, so if you publish, okay, so ours is human after genome human genome project, we got 3.2 billion base pairs. So if you print one book, okay, so this is one book, so 1000-page size, okay, 1000 page one book, so into 1000 books, so that big our, okay, so 3.2 billion base pairs of, so which is derived from 20 to 25,000 genes, okay, number of genes. So now, okay, so you all know this. What is this? It is a Flipkart homepage, right? So whatever you want, you can order there. So order it now. So what do you want? Two-wheeler. So definitely you will get. Okay, so e-bikes are now provision is there. Okay, not only this, they have made MOU with Hero and other companies. So you will get it, right? So what they will do? So they will, okay, the information you will get. So why I have just included this? Because so behind the Flipkart's efforts, so to reaching out whatever you have desired the product, either you take smartwatches, either you take a book including pen, whatnot. So all the information you are going to get. So it is all because of maintaining very big, okay, the warehouses. Okay, like this, the biological data system also having warehouses. Okay, so warehouses. See, this is spread across 110 acres as 5 million cubic feet of storage. So look at the capacity. So that for that reason, so they are catering and they are very fast in delivery, right? And data warehouse. Okay, so how it works? See, data sources are genomics, proteomics, and metabolomics. So whatever the information is you have okay, gathered, so that will be deposited here. So what is that called as data repository system? Data repository system. It is just like your saving bank account. Okay, so whatever you earn, whatever you are earned, so that money will be deposited there. Similarly, so whatever the information you have worked in the laboratory, so that will be deposited here. So that is called as metadata repository. Okay, so metadata repository. So what the imaging and also the physician researchers, what are all information related to the biological science, so that will be deposited here. So this is what we call as large scale data facility. So what you will do, the servers will be there and servers will take care of this. Compute system access and HPC clusters, and researchers whatever they will do, so they will feed it here. And cloud storage, okay, Google Drive. So if you go anywhere in the world, so you can access through your documents, so whatever you require. The similar pattern, so this also it works. This is where okay, so the sharing of information, you just keenly observe these arrow marks. Okay, so how the data system okay, from the data warehouses, the mutual exchange of information takes place. So database, okay, so is a vast collection of data pertaining to a specific topic. Okay, so depending upon your interest, so that you are going to gather the information. Again, it is a 10-mark question. So what is the biological database, and explain in detail. Okay, so database is vast collection of data pertaining to a specific topic. So example, nucleotide sequence, protein sequence in an electronic environment. Okay, so if you print it in your department and keep it, so it is not publicly available, so it is not in the public domain. So there are heart of bioinformatics, these databases are and computerized storage for data records, allow extraction of specified records, allows adding, changing, removing and merging of the records or the information which it is available. Use the standardized formats. See, for biological data students, so you cannot use .word file or .xml file. So what you need to use a FASTA format. FASTA format. So what is the so in FASTA format there are no table, no images, no photography, nothing. Okay, so only linear alphabet model. So what is linear alphabet model? So only ATCC will be there. So nothing else. So where into the some of the formats. Clear? So but for what about the protein? PDB format. Okay, for protein data bank format. Okay, for protein structure deposition as well as for protein sequence. Clear? So any doubts here?
No, sir.
Okay, so how you need to access through your system? No. So you cannot do it. So for that you need to install a software. Okay, FASTA reader. Okay, FASTA format reader. So different types of database, I will just go through it. So enzyme database, so where you will get, so here. Okay, Brenda. And disease database, OM, IM. And chemical database, PubChem. Microarray database, GEO. Taxonomic database, catalog of life. And bibliographic database, PubMed. So most widely used. So I am just mentioning it in reversing order, but it is the first one. So where the scientists across the globe they will use. Okay, sequence database, GenBank, DDBJ, PIR, and also EMBL. So there you are going to get. So these are all the name of the databases. So whichever you want, you can just type in Google. So you will get the information. Okay, PDB. Next one is metabolic database. So if you are really want to work on metabolic disorders, so definitely you will get the information here, students. Okay, so for that you need to no need of acquiring the knowledge of SQL as well as the Python programming languages. Okay, so now in Udemy and also NPTEL, so you will get all this information free of cost. Okay, so you can learn, so you will become a successful bioinformatician, right? And also model organism. Okay, so one is given here. Okay, so can you name one or two model organisms? So Arabidopsis thaliana is one in plant, E. coli, Drosophila, Melanogaster. So all are called as model organism. If you want to take a data of rice, so you will get here. Okay, one more database is the plant. Okay, the name itself plant. Or if you want a database of related to different species of rice, Orizae. Okay, database. So you will get all the information related to this. nucleotide sequence databases. So important one. Okay. So three different database set are there. So first one is NCBI, Gene Bank, EMBL, DDBJ. So where okay, so DDBJ is maintained which country? Why? So which country DDBJ is maintained? It's all about in the J only. Japan. Okay, right? EMBL, what about EMBL? Which country?
Europe.
Europe, right? And what about NCBI or Gene Bank? USA, right? Okay, so these three databases are updated and exchanged on daily basis, and the accession numbers are consistent. See, if you want to deposit, now you have done some lot of work, okay, into the plant sciences, and you have come across some sequence of some gene, right? So that you want to deposit there. So for each depositions, you are going to get an accession number along with your name and institute affiliation. Clear? So with your name and also with your institute name which you want to affiliate, so you are going to get the accession number. What is accession number? What is accession number? So it is just like the USN number, university register number, right, or roll number. So okay, so no need of pointing you through your name, right? So what you can do, easily if you type the USN, what is that? Accession number. So easily you are going to get the information, students. Okay, so there is no need of remembering everything. So if you just remember the accession number and just copy paste, you will get the same information. So there are no legal restrictions in the usage of databases. However, some patented sequences in the database. So if they are not patented, so easily you can download and you can utilize the data however you want. The international, already I mentioned this. So because of the collaboration, it is made available to all. So this is the home page of NCBI. So today, so to understand okay, the concepts of bioinformatics, go home and Google it. Okay, so the URL is ncba.nlm.nih.gov. So if you do that, so you are going to get this page. Okay, so here that drop box is there. If you drop down, so what you will get? You will get the information of gene, SNPs, and nucleotides, whatnot. So all the whole genome organization, all the information. So you are going to get. So what you need to do? So already mentioned accession number. So enter your accession number. So that is what I have shown you Flipkart home page there. So whatever the item you want, you can type it there. So now if you type Tri1AC or BRCA1, so what is that? A breast cancer causing gene. Yes or no? BRCA1 is a breast cancer causing gene. Okay, DRO1, it is a drought resistant gene. Okay, so if you insert DRO1, okay, for the invention or okay, for pointing out the DRO1 gene, Japanese scientist, he got Nobel Prize in the year 2020. Students, he got Nobel Prize in the year 2020 for just to identify which gene is responsible for showing drought resistant among the rice plant. It is a Japanese scientist, he got Nobel Prize for the same. Okay, and if you type here, you will get all the information. See here. Okay, so in the home page, chemical and bioassays, data software, RNA, domain structures, gene expressions, genetic and medicine, homology studies, proteins and sequence elements, all the information you are going to get. Okay, so through this. So which one? NCBI. Okay, so national. So who is maintaining it? So National Library of Medicine. So I will explain at last one more point is there. So why PubMed is okay, so largely used. Okay, so this PubMed is why it has been largely okay, utilized for the studies. So next one is EMBL. So all of you know that. Okay, so when it was founded, so which year? 1980. Okay, so at least you remember. I am very happy that you are following, right? So European Molecular Biology Laboratory, EMBL. Okay, so EMBL means European Molecular Biology Laboratory. So maintained by European Bioinformatics Institute, that is EBI. So what it will do, it will give genome survey sequences, high throughput cDNA sequences, high throughput genomic sequences, express sequence tag, as well as the patents which they have taken for their findings. Okay, so this is the service, so what they give, and exactly similar to the NCBI. So what are all the features? It is available, the same set of information also available into the EMBL. Okay, but here one added advantage is that, so along with the nucleotide sequence, here you will get the protein sequence also. Okay, and also you can generate, you also generate three-dimensional images of the proteins. So this is what how the EMBL home page it look like. So this is the URL www.ebi.ac.uk. So what is that UK.uk? It is a country domain, and if .com is there, so it is globally available. Okay, the information. Okay, so like this. So next one is the DDBJ. So developed in 1986 as a collaborative with EMBL and GenBank. So produced and maintained, distributed in National Institute of Genetics, that is NIG, Japan. So sequences are submitted via web-based data submission tool. See, so if you are done with this any work, and if you are having some set of data, so definitely one can take the assistance of the professors. Later on you can submit the data. So your name also will be recorded over there. So this is the home page of DDBJ Japan. Okay. So all the information in search tool. So here, if you are able to retrieve the data from the database. So what all information it is required, so that information you are going to get. Submission. How to submit your data? See, these the website interfaces are very user friendly. Okay, so if you know proper reading, English reading, so you can easily achieve whatever you want, because video tutorials also available. Okay, so along with this, video tutorials also available. And what are the services? See here, many of you don't know. So the internship opportunities also there for graduates. Okay, so you need to visit this database, and so you may have the chance of working at DDBJ Japan. Who knows? Okay, and also EMBL also. Okay, so what is that unique property you need to have? You need to have first interest. Okay, so interest, so it will grow. So if you make it possible, so definitely, so one of you are sitting here, can work at DDBJ Japan. And here the specification and statistics and activities done by DDBJ, you visit activities. So what are the activities they have done? Okay, for past year, financial year, or whatever you say, academic year or whatever, so related to the agency, so that information you are going to get here. And other databases also there, students. Okay, so these three are there, so they are very much important. So which one? First one is NCBI or GenBank, next one is EMBL, and also DDBJ. So EST is express to sequence tags. For that again separate stream of database systems is there, so that also you can access. And it is also called as GenBank subset. So with additional EST specific data. So who are looking for express to sequence tags information, so exclusively you can visit here. And SNPs also. So SNPs are the new future. Okay, so because already I mentioned why you are different from your friends, because of SNPs. So how best you can manipulate them, and how best you can use it for the development of new drugs as well as medicine, that you can explore them. And many commercial databases are there. Silera, one example. Commercial databases are there. So there, the rest of the domains are publicly available, public domains. So these are all paid ones. Next one is in biological database, protein databases. In the protein sequence database, so they will function as repository of raw data. Two types are there, primary as well as secondary information. So what is primary data here? In the protein sequences, raw data. What is the raw data, students?
Whatever the research we have done without any confusion.
Exactly correct. But in the form of again the linear alphabet model. Okay, amino acid sequences. So that is called as primary data. So from primary data, you need to generate some three-dimensional structures, right? Okay, for where it is used, whenever you develop the new drug, so there it is most required for the docking purpose. So at that time, so you will go switch for secondary sequence database, okay, or secondary database. And the next one is the protein structure database. This is how okay, so the hemoglobin, three-dimensional structure. Hemoglobin is what? A protein. Although, yes or no?
Yes, sure.
So can you name some of the proteins? So which you know?
Lysine.
Proteins.
Lysine, arginine.
How many of you eat egg morning for breakfast? Albumin. And the BSA experiments Boeing serum albumin. So you will use, right? So for most of the experiments, correct. Hemoglobin is one. So it is present in blood. And keratin, nail and hair formation, right? Collagen. So many proteins are there. So those studies you can do. So where? So here protein structure database. Okay, so this is one student see here. Okay, the 20 amino acids are arranged in this format. So I have taken this for your reference. Okay, so this is hemoglobin. Okay, so all the information are given here is the first name of the amino acid. Alanine is there, right? So likewise it goes. So UniProt is one. Okay, when you visit PDB, okay, protein database, so it will redirect you to the another subsidiary of PDB. So that is UniProt. Okay, so where these okay, the repository of protein databases are present. Okay, so again, okay, for your reference, I just wanted to add here because if you are having confusions, it is very difficult to understand. See, you know this information, right? Replication, gene DNA, transcription, and DNA to RNA, messenger RNA, translation, and so this is what the one first. What is that protein? It is a combination of amino acids. Okay, so these amino acids you will get it through the sequencing. Clear? Any doubts so far? So related to this, how you are generated? So for getting protein sequences is different. You are not going to use Sanger method of sequencing, right? So this is how the protein data bank, the home page look like. Okay, see 2,06,239 structures from PDB. Okay, so they are deposited and also they have generated around okay, so more than 10 lakh computed generated or structure models here and here. So the deposition is there. Whoever see one of my okay, lab mate when we are when I was pursuing PhD, so the research. So he is working on this silkworm. Okay, so the saliva or the vomit. So whenever he used to treat it with the chloroform, it used to omit some self-defense mechanism, some like saliva kind of okay, the liquid it used to spit. So at that time, so he has found that it has some proteins, so which is most suitable for the treating the cancer, different types of cancer. See, such type of studies, he has sent the sample for the sequencing and he has published into the protein data bank. Okay, that information he has published. So next time, whenever you want to use that, okay, the information, so easily you go here and search. Okay, the entry returns, entry IDs and sequence, same accession number. However, whatever you have done with the nucleotide database, the same information, same methodology you need to adopt here also. Clear? Any doubts there? Okay. See here, experimentally determine the 3D structure from protein data bank. See, only one constraint for this, you cannot perform all this with the old age computers. So for this, at least you need to have i5 and also some SSD. If it is there and also high speed internet, so then easily you will get. And whenever you are planning for working on human proteins as well as human nucleotide sequences, the base pair number is very big. So the loading itself it takes the time. So we used to do okay, the comment on this. So whenever you used to load, for example, BRCA1 breast cancer one gene. So if you load it, so you can have your lunch and come back. So by the time 20 to 30 minutes, so it used to take place to load, okay, the full nucleotide sequence. So that is how the things used to work. But now advancement with the okay, the engineering as well as the computers also upgraded. So now easily you will get okay, high throughput 5G network. Okay, so these are all primary databases under protein, right? protein databases. Swiss Prot, the groups at Swiss Institute of Bioinformatics, that is SIB. So if you want to pursue your masters, so definitely, so these will help. Okay, so you can note it down. Okay, so after your okay the graduation, so definitely you can okay so access all this information and definitely you can achieve better rights, right? So it announces the okay the sequences, describe the protein functions, its domain structures, post translation modifications, after translation what are the changes okay into the protein. So that information you are going to get here, and provide a high level of annotation and the minimum level of redundancy. Redundancy is see whenever you perform the work, okay, so after a week or 15 days or 1 month or a year, so you need to able to reproduce the same results. Okay, so you need to able to reproduce the same results. If you are getting something else, so what it is called? It is not reliable data. It is not reliable data. You have done some mistake. See, at the very first point of you are getting the results, you cannot go and publish. So you need to reproduce it again and again. So that is what we call as redundancy of data. So that need to be there. Means very less redundancy levels need to be there. High level of integration with the other databases. Thus, these are the some of the salient features of this Swiss product. And PREMBL is there. So again, it is one of the subsidiary of EMBL, European Molecular Biology Laboratory, and computer annotated supplements for Swiss product that contains all translation of EMBL nucleotide entries. So they are not yet integrated into the Swiss product. So which are all the lacunas are there, so that you will get. It is just like which are all the products you won't get in Flipkart, you will order in Amazon. Okay, so same pattern. Okay, so TRML as well as the Swiss product they will work. And next one is the PIR, Protein Information Resource, a division of NBRF in US. So collaborated with Munich Information Center, and what they will do, so one and okay search for entries and do sequence similarly as well as, so it is able to produce the database of sequences extracted from 3D structure in PDB, okay, protein data bank. So this is the home page of Swiss Prot. Okay, so same. Okay, the blast is basic local alignment for your sequences. So whichever you will get. So whether any similarities are there. So in the earlier of this talk, so I just mentioned you. So if someone if they have done the work, okay, so with this, so you need to relate it. Okay, the same sequence has been related, and that you can okay, so get it here. So one more important tool is there, Blasta. So that we will okay discuss in the later part of the classes, right? And the secondary databases are these ProSite. So what they will do? So they will process the basic primary data and they will give you the structures. Okay, the most refined form of data they are going to get. It is just like you have done BSc Biotechnology. So you will become more refined in this subject when you join MSc. Clear? The similar kind of information regarding proteins. So you are going to get in this secondary databases, right? See here, the secondary database compile and filter the sequence data from the primary database, and these database contain information derived from protein sequences and help user to determine whether new sequence belong to the known protein family or not. So here you are going to get the information and you can draw a conclusion whether it belongs to new protein family or already it is presented in the repository. Ad go down it will check. So if it is there, so it is of no use, right? And such type of secondary databases are ProSite. So the database of short protein sequence patterns, profiles, and characterize biologically significant sites in the proteins. Very important. The ProSite is one okay the tool which helps in characterizing the biologically significant sites in proteins, and also it gives you regular expression studies and characteristic sequence of specific protein families as well as domains. It is part of Swiss Prot and maintained by the same way. And next one is the prints. So it is provides the compendium of protein fingerprints. Okay, so when see already some services are there. So genomic studies. See, for example, if you want to know your ancestor, so that can be achieved here. Okay, so the scattered population, how many around 7 billion population is there in the globe, so you can draw a phylogenetic tree. Okay, you know what is phylogenetic tree? Phylogenetic tree. Evolutionary relation. Okay, so among the species. Okay, if you draw evolutionary sequences among the other animals, so who are our relatives? You know. Okay, so if you draw evolutionary sequence, okay, amongst the human and with whom we are sharing. So who are those? Apes, or you can call, okay, chimpanzee or gorilla. So they are sharing 99.8% of genetic similarity. Okay, so between, okay, these two. So next one, blocks. So these blocks patterns without gaps in the aligned protein families defined by proteins. And so what they will do? So they will help in conserved segments. And TFAM, the database of protein families defined as domains. For each domain, it contains multiple element of set of defining sequences and also others. Okay, so in primary, in a protein database, so two types are there, primary as well as secondary. So these are all the examples for secondary database. You just remember prosite, prints, and blocks and PFAM. See, bioinformatics is one subject. Okay, so if you are having after this session, when you go back home or tomorrow you sit into the digital library, so just access. So what are the okay, the website address which I have given. So if you just once if you explore, so all the information definitely it is not going to wash away. Okay, so then that is what okay, so the practice you need to make. Okay, the practical sessions need to be there for bioinformatics, right? So you need to do. So for that hands-on training is required. So for that what you need to do, you just do okay, some trial and error method. Okay, so take one gene name and put it there and try to retrieve the sequence. One I will show it. Okay, so after this completion, so I will show how to retrieve sequence from the database. Okay, so how sequence, how does that sequence look like? So these are all again protein structural database. PDB is there. So here you are going to get 3D structures of biological macromolecules. So how you can okay, deposit these 3D structures of okay, so the biological macromolecules through X-ray, crystallography, and NMR. So, into this domain, students, listen, please concentrate here. Okay, so PDB, so for obtaining the 3D structures, so what you need to do? NMR. So here NMR andre abbreviation.
Nuclear magnetic
Okay, so
resonance.
nuclear magnetic resonance. So that you know, aware of. So you need to do that. Okay, so you need to have a hands-on training on how to use NMR. See, since you are under the okay, the magnetic resonance, so you need to be very much careful, right? So all these precautions need to be taken care of. And X-ray crystallography. Okay, so only very few sophisticated laboratories are there. Okay, so laboratories are there. So they will give this facility. So one such you will get in IASC and also in NCBS. Okay, so you are going to get this information. The only thing what you need to do, you collect the information and submit there. So they will give you the 3D structure of protein. Macromolecules means proteins only. Okay, don't get again confused. What are these macromolecules? Don't scratch your head. Right, PDB entries contain atomic coordinates and some structural parameters connected with the atoms and computed from the structures, that is the secondary structures. The PDB provides primary achieve all the 3D structures of macromolecules such as proteins, DNA, RNA, and also various complexes. So that information you can deposit in your name, right? And this is a scope, SCOP. Okay, structural classification of proteins. The database was started with the objective of classifying protein 3D structures. See here, no classification of features are there. Okay, so you will, whenever you want to purchase a new cell phone, so you will look for the features. So what is the megapixel, 200 or 500, right? So and you will look for the storage and you will look for the processor, which process is there. So similarly, in a single phone you will get all the features? Definitely not. So if you purchase Apple, so always you need to carry a power bank with you or a charger. Correct? Yes or no? Right? So if you buy any Chinese manufacturer, so you need to always okay, so fight with the ads which are coming in front in between. So when you are working with them, right? They are called as bloatwares. So for that reason, so why I am explaining all this, because So you will ask, okay, so next point after this completion, sir, why that protein structure database are there? So why so many the domains are there? So why we need to study all this? So this is because of, see, some are having, okay, some limited okay, the features, and some are having added one. Okay, so alva, isn't it? The purpose is same, but the thing is, some are well organized, some are not well organized. See, that is what here. Okay, the classification is not done here. Okay, where, where? Okay, in PDB, so there is no proper. Okay, the classification. So here it has been done. So it is based on the data in primary database, but adds information through the analysis and organization. The classification. Okay, so that is what we call it as hierarchical scheme of words and superfamilies and also families. So how we were okay, the classification in botany or zoology, the classification is done. The similar way here also the classification is been done. So this is again students. So one is okay, so this is the protein albumin. Okay, albumin structure, 3D structure of albumin. See, look at this. It is very complex in nature. This is the 3D structure of keratin protein. Okay, so it is just for your reference. I just wanted to show you. So how the 3D dimensional structures of proteins they look like. Okay, so when you next tomorrow morning when you see a egg, so you need to remember this structure, how complex it is in nature, right? So this is wonderful. Okay, the software students catch. It is called as class architecture, topology, and homologous superfamily. So this is again where you are going to get the information about the classification of protein structures, so which are downloaded from protein data bank. The protein domains into the superfamilies when there are sufficient evidence, so they diverge from the common ancestors. So the here the classification also is based on the ancestors, right? So already when we are discussing about the hierarchy or the pedigree analysis, or when you are drawing the ancestral roots, right, or a tree diagram, so this will come into the picture. So what it will do? So it will help you the Gen 3D. So this is also one of the tools. So you can note it down and you can practice it later. So with this tool, okay, so you could able to see okay, the many okay, the improvements are there. For example, in how you are in a lens card. Okay, so when you just scan your okay face, so it will give you so how you look into the new spectacle, right? The similar way in catch, sorry, in the gene 3D. So you are going to predict the structural domains of millions of proteins that are available in the public domains. Okay, so that information you are going to get in gene 3D. Okay, it is also one of the best okay, interface for understanding the three-dimensional structures of proteins. So which one? Which one? Gene 3D. Okay. So additional annotations to the catch and gene 3D database such as the functional information and active sites or residues. See here one more unique feature is there. So in this, so what you will get as soon as you will type a protein name, so what will happen? So you will get the where exactly this okay, this protein is present, what is the three-dimensional structure, and also it will help you to get the functions of it. Okay, the functional properties. What exactly this hemoglobin will do? So what is the function of hemoglobin?
Is to carry out.
So it will supply the oxygen, right? Albumin. So homework for you. Okay, so please note it down. So what is the function of albumin in our body? See, albumin means it is not only present in egg. So it is present among mice also, rabbit also, in many organisms. This albumin protein is present. Clear? In humans also it is present. Right? Students? So you need to find out. Please note it down. So albumin. So what is the function of albumin? Clear? So you need to. Okay. So next when we meet in at online, so definitely I am going to ask you. So please answer. Okay, for the same. Right? And also you write. Okay, all of you. So what is the database and submit it to sir. Okay. So what are database? What are biological databases? So questions for you. Okay, 3 to 4 questions. Okay, so how bioinformatics, what are applications of bioinformatics and opportunities of bioinformatics. So please note it down. All of these are assignments for you. Okay, or questions for you. So practice sessions, right? So what is NCBI? Abbreviate as NCBI and describe the salient features of the same. And what is EMBL or abbreviate EMBL? Write the functions of EMBL. So what is the DDBJ? Same. Okay. So what is the functionalities? And what is the PDB? Abbreviate the PDB and write its salient features. Okay. So what is primary data base and secondary database? So in case of PDB or protein database. So what is the catch? CATH.
NAS architecture.
Okay, so that also you need to write. So what is enzyme database? So that information also you need to write. See, all this information I will provide. So only thing is you need to reproduce in your assignment book. Clear? Okay. So next database is enzyme database. So it is brown schematic enzyme database. So it is abbreviated as Brenda. Okay, so BRENDA, right? So it is an enzyme, a part of SY. So that is Expert Protein Analysis System, the proteomic server of Swiss Institute of Bioinformatics. So why enzyme data is very important, student here? See, enzymes, you know, okay, so they are what enzymes will do? They will increase the rate of reaction, right? Or they will accelerate the rate of reaction, right? So what is the use? What is the use? Faster the you are going to recover the product in faster way, or time is money. So in industries, so as soon as you will get the product, so that will be purified and it will be sent to the market. And meanwhile, see the enzyme data why you require, because so whenever you set up an industry, any industry or food industry or beverage industry, so you need to look for the quality, right? So without quality, it is not going to work for you. You know ID. So what is ID? Idli dosa batter. Correct? So you know the story of ID, how Mustafa, he was dropped out from 12th standard and he started with a small business, 500 rupees in his pocket, and now he set up an industry of 5,000 crore worth industry in Bangalore. Okay, so he was roaming into the streets of Bangalore without any footwear. So now he set up an industry. Okay, so with 5,000 net worth of company. So what is that company name is? ID. And okay, so he didn't stop there, students. He start okay, what he did, and the vada maker also. See, you don't have, see, Bangaloreans, so you don't have enough time to prepare the batter and make his vada. So what he has done, he designed a cap in such a way that he has having 2 patents on only cap, not on the product, only on cap. If you squeeze that, so you will get exactly the shape of the vada. So you will get into the MTR or the Mawadi tiffin room, you call alva, Brahmin's cafe bar. How you get the same, exactly the same composition and taste, you are going to get that vada. Okay, so that is what you need to think. So it is not that only you need to perform this and to do it. No. Okay, so never stop at okay. So for your subjects. Okay, so think out of context and you will be successful person, because we are 140 crore population. So whatever you produce, manufacture and cater to the people, they will consume it if it has quality. Okay, the people are there, they will go to Vaskote to eat Dhanne biryani early in the morning, 4 o'clock, 4 a.m. Even they are not brushed. Right? Correct? So why they will rush there? Because of quality. Okay, you do with the passion, you do with okay, so all this, so you are going to achieve some of the heights. Now 3D printers are there, food printing machines. So you can print dosa and idli. Now ATMs have been already established, right? So if you want masala dosa, open bender dosa, so it will give you. Correct? So they are all innovations. See why I am stressing on this. See, ID is the success formula is it won't get so early. Okay, so yeah, he used to give in a plastic pouch earlier. So now what has invented? Ziploc pouch, because okay, at now new age women, so they have no time to wash the vessels. Correct. So 3 doses if you want, okay, pour it and put it into the zip cover and throw it into the refrigerator. Close. So next evening you come and do the same and keep on doing the same. Okay, but what is making the difference? ನಿಮ್ಮ ಮನೇಲಿ ಮಾಡಿರೋದು ಹುಳಿ ಆಗುತ್ತೆ ಬೇಗ ದೋಸೆ ಹಿಟ್ಟು ಇದೇಕಾಗಲ್ಲ because of see preservatives are there. Okay, but the thing is the enzymes which they have used, okay, in the form of yeast, so that is very important. Okay, so one of the conference I attended at Bangalore University Granberthy campus, one of the added used to say, okay, so the scientist is from okay, the Food Corporation of India, so he has patents on some enzymes, food producing enzymes. He is earning crores of rupees. Okay, he has developed, okay, and he has proven, so it is giving a 10% extra. See the product yield. See companies what they will do. So if you are spending okay 1 ton of raw material in the production, so the losses will be there usually, okay, as a waste product. But okay, so if you utilize that also and if you give 10% extra, so it is the most profiting business for him. For whom? For industry, right? So you need to think in that context. See everything, it is not going to end. Okay, the continuous development and innovation, it is not going to end soon, because okay, in 2005 Android, okay, the generation first, now 13, and a lot of evolution has been taken place, and new software, new tools and new features, right? So if why Nokia failed? Because they are hesitated to update. Okay, so they never come in contact or okay, make a MOU with Android. So they are just to stick to the Symbian operating system, and they are just to stick to the MS Office. So for that it is not a user friendly, so they are just vanished from the market. You are understanding? So you need to keenly observe, so what is happening in and around you, so you will get the idea. Okay, so that we will talk in research methodology. So these are all okay, so clinical databases we will discuss in okay, other session, students. So something important is there, so that I will cover. PubMed. Okay, so where you are going to get the okay, the okay, high quality journals, okay, the publications. Okay, so this is where you are going to get, okay, PubMed. See, whenever your professors they say, okay, so get the reference for this, so this is one platform, so where you just give the keyword, so you are going to get the very quality journals or the research articles where in PubMed. Okay, so this is how it look like. Okay, the international corruption and okay, so that information. See here one more thing, free full text is there. If zero symbol is there, okay, students, you will get it for free. Okay, some are there, okay, the paid version. Okay, so paid versions also there, that you can request letter also you can send to the author. So if they are willing to send, so therefore definitely they will send. The way of asking need to be better, otherwise they won't send. Just send your article, no one will send. So why you need it? You need to write in a descriptive manner, so that you are going to get the reply. Okay, so the future of bioinformatics challenges, data value. Okay, so the growing data. Okay, already mentioned. Okay, how many? 65 lakh crore database already into the system. So we need more space for the okay, more space for the data accumulation. Okay, the issues this will cause the data management in terms of storage, transfer, processing. In addition, data integration will be the key challenge, and data access, another big challenge. So you don't know how to access the data. Most of you don't know how to use the keywords. So why the keywords will be there? Why the keywords will be there? So when you click a selfie at okay, so Pizza Hut or Domino's, so you will put a hashtag there. So they are nothing but keywords. So that when next person, so whoever is there, when they are scrolling, your photo also will come. So it will increase your popularity. The similar way, the keywords are very much essential for increasing the reach of your work. Correct? So to reach your work, see, you need to relate and you need to think in virtually Okay, whichever the way you want. Okay, so then only things will be easy to understand. So otherwise, whatever I am saying, if you are stick to my wordings, it is very difficult for you. You stick to your which is your favorite one. Okay, for example, social media. Okay, so similar way the keywords also follows here. And data access already I mentioned. See, this does not help the scientists familiarize themselves with this data type and value it can add their research. And hopes for future bioinformatics. The creation of more machine learning based discovery methods. Already in engineering courses are there. Artificial intelligence and machine learning. Okay, so for example, one example I will quote and I will move on. Okay, so in a field, okay, if you want to pluck a weed, how it will be knowing? So whether it is a weed or crop, main crop. Okay, so machine learning, you need to click a thousands of photos and you feed it, okay, into the machine, so it will learn. Okay, oh, this is weed, I need to pluck it. Okay, for example, Parthenium, how it will go and pluck the Parthenium, because see, the funniest thing of our India is, okay, so we are 140 crore population, we don't have laborers to work at the fields, and they need employment, right? No laborers are there in villages. All the villages are becoming orphanage or home age people, okay, or old age homes. Only the old people are living there. If you go Malnad region, Shimoga and Chikmagalur and other places, so no, all youngsters are into the here they are working IT or BT somewhere here, or they are becoming a city boy delivery or whatever. Okay, I am not blaming the condition, but this is the fact. Right, correct. So all the people you ask, okay, next time when you get the delivery, so you ask which place you belong to. So they will answer you. Right. So all are becoming because the village guys they are not getting girls for marriage. Right? Okay. So this is the harsh reality because okay, the generation it is over brainwashed with the social media. Okay. So the over brainwashed with the impacts of social media. You want to lead a life in such a way that your friends does. Okay, you don't. So that is what again the comparison between you and your friends, and you want to go to US only. If you are married and you want to US, so I want to go somewhere else or better country than Earth. So such a type of mentality. So the boys in cities, if he is doing okay, so here delivery boy or whatever, so the girl will marry because he stays at Bangalore. Whether he washes the car or whatever, so he stays at the Bangalore. So this is the condition. So the village guys are facing students, right? The creation of more machine learning based discovery methods. So this is very crucial. Okay, this is very crucial. Okay, you need to address and the more usage of genome-wide associations to understand diseases leading to more therapies for patients. Okay, so again, see the future of bioinformatics. So if you are having proper data, so you can cure the patient with the precision medicine. Okay, precision medicine. So you can cure them. And wide adoption of high resolution sequencing methods. Okay, so better the resolution, so better impact will be there. So what are the at last? Okay, so it need to become first because so without okay the way you will reach at the end of okay learning bioinformatics. So here you will okay reach or here you will join. See top 10 job opportunities in bioinformatics in India. Since India is very emerging and okay the glorious okay duration. Okay, so you need to make a path. See, none of the institutes are making okay, so this, and they will tell you. So if you do this, you will become this. Okay, so none of the education institutes are there because our education system is like that. Okay, right? So in Netherlands or in Australia, a plumber will get more money than any of the professions. Okay, because all the professions are seen equal. So here it is not going to happen. So for that reason, we are under the fantasy. If you do computer science, you will earn 5-digit salary. So that impression we are all right. And see, bioinformatics software developer. So first job criteria. So bioinformatics software developer work on the development of existing software tools and analytical skills. What you need to have? You need to have a analytical skills and good communication skills. I think all of you have good communication skills here. Who are sitting, all of you. So to be able to create new tools and new algorithms, it is not very difficult, students. Okay, if you refer some of the video tutorials and some action trainings also will be given personally. If you spend some couple of thousand rupees, so definitely you will also become a developer for new algorithms. Means algorithm means already I mentioned. It is a what is algorithm? What are algorithms? It is a solution. Okay, solution based concept where you are finding a problem and you will solve. Okay, so that is what algorithms are called. Okay, so that need to have a good knowledge of essential art skills such as SQL and Python programming language. Already 3 times I have mentioned here. Okay, so if you learn so these programming languages, it will be easy for you. Companies building computational tools mostly are bioinformatics software developers. So if you are having, see what computer science graduates they do. Okay, so they cannot do and they cannot analyze biological data. Okay, but you can, if you learn these two, it is very easy for you to be a bioinformatics software developer. Clear? So now who is working? See, one next to the software engineer, so one graduation from biotechnology need to sit because you don't have software skills. He doesn't know what biotechnology is. Right? So if you understand and learn these tools, so it is not very far. So you will become a software developer. Companies, okay, so they will hire. So which are all the companies they are hiring? So Philips, so tube lights, okay, light system, you know. So they are having healthcare wing, okay, and also Wipro, okay, Infosys, and almost all the majorly the Biocon, okay, so in biotechnology. So they are all having this separate wing of healthcare wing. So they are working on it. Okay, so why they are working on it? Because it is a future emerging field. Okay, so healthcare, it is the biggest okay, so marketable, okay, in the near future, students. Okay, so next one, bioinformatics research scientist. So for this, okay, so you need to okay, so use computational methods to study the living system on the earth, and based on the data sets outcomes and okay, the computational methods, they found the answer to provide the new information related to the field of biology. See, you don't do anything, collect the data from various sources and analyze it. So what is happening? You try to relate with something else. So that is what you will become bioinformatics research scientist. See, bioinformatics research means it is not that you need to send some sample for sequencing. No. So what is already existing? So you need to okay, conclude. So already you mentioned, right? So only the data is available, raw data. Okay, so many scientists what they will do because of their economic background and because of their many family issues, so they cannot continue okay, and put a full stop for the work. Okay, they won't conclude. So such a type of work you can carry out. Okay, so without any hesitation. Right, after pursuing PhD in bioinformatics, a bioinformatics scientist can take up several research positions in and around the many institutes which are present across the globe. Right, next one is bioinformatics analyst. Bioinformatics analyst studies the complex nature of life chain, conducting research in laboratories, developing information algorithms, creating database that can help the future analysis of several diseases and physical conditions of the different types of different okay, sorts of diseases. Okay, so which people are suffering. To some extent, bioinformatics analyst is similar to bioinformatics scientist. These professionals explore career in okay, so bioinformatics after pursuing master's degree or PhD. And bioinformatics engineer, they are the professionals who are responsible for creating the computational methods. The same set of work you need to do. For that, what you need to have? You need to have a good knowledge of database. So, what is database and what is the data subset? So that information you need to have. Okay. And the computational biologist. So the best bioinformatics job for freshers or recent graduates. So if you type this in naukri.com, okay, or Times Jobs, so definitely you are going to get many of the okay, opportunities. And also I think all of you heard about medical coding jobs. So the medical coding, it is also emerging trend. Okay, so and the students are getting starting package starts from 5 lakh rupees. Okay, per annum. Okay, so it is approximately around 45 to 50,000 rupees. Okay, 5 lakhs package. So soon after completion. So for that you need to again I already mentioned. So many courses are there students online which are free of cost or some are there paid one. So that please you okay, do it and look for the other alternatives. Okay, so don't be in a crowd and don't follow the same set of traditional job patterns. And the database programmer, okay, the computer programs and updates them. So you are the one, so you will update. See, now I already mentioned. So you developed, okay, so generated some sequence and published it. Who is going to, okay, manage it? So you are the one. So you are the database programmer. Next one is bioinformatics. So they will work closely with the biologist and geneticist. See, in usually in medical colleges, the research wings will be there. So there he will get these opportunities. Now it is mandatory. Okay, they need to set up a research laboratory in a medical college premises itself. So there, so definitely you are going to get this information because the physicians or the doctors, they don't know the ABCDs of bioinformatics. So they will just give you the data. You need to compile it and you need to do the do the half work for them. Okay, so they will again drive conclusion. Next, freelancer. So it's not that you need to so work in one premises and if you are not interested into the 10 to 5 job pattern. So definitely you can think of freelancer. Both academic and commercial divisions. So they will help you to compile the data and you will become a part of freelancer in bioinformatics. And the core facilities, so national laboratories, so there they will be having their own facility. So there also you can join. For example, already I mentioned NCBI you can go and who are doing research in life sciences. So there you can definitely you can join. So last one is educator. So what I am doing now? Okay. So this is the last one. And making and teaching you all the top 10 so opportunities, so which are all in the bioinformatics. Okay, so for this what you need to do, bachelor degree, masters, PhD and so on. Okay, postdoc. Okay, and it is a never ending story and you need to update yourself to the present context, right? So this is all about it is our college, Biotechnology. So it is at present at central part of Karnataka. Okay, so this is not the end. This is just a introductory. Okay, class. Okay, so I think I may be very fast to reach this last slide. Okay, don't feel that. Okay, so next time whenever we interact, so what are the issues you are facing, you can ask. Any doubts there, you can please ask. So this is what we will discuss in coming classes. Database main tools. Okay, so how does these database main tools are used? Okay. Basic local alignment search tool and homology analysis. Any doubts, students? One or two? Clear? Now I will ask questions. What is database? Now I will ask. If you don't know, I will ask the questions. What is database?
A database is a vast collection of data from a specific topic.
So that is what you are doing, that is database. Okay, so collecting attendance or information. Okay, so but don't write any form of information. Since you are a biotechnologist, please mention the wordings of it is a biotechnology or laboratory generated data. So which is stored in the databases or servers. So whatever. Okay, so that is what called as database. Here, NCBI full form. One second. You are not playgroup students. If I say, please tell Jolly Jolly Escobar. So, okay, don't be in the chorus. Please look at your notes and tell. Sit and tell, not a problem. Very good. Where it is situated? EMBL full form. One second. I already mentioned you are not a playgroup student. Sit. Please look into that and tell.
European Molecular Biology Laboratory.
What is DDBJ last? Done. Sitting over there. DDBJ. Please look into your note and tell. So we will put it in audience poll. Audience poll or phone a friend. So which one you will choose? Audience poll. Yes, please.
Japan.
Okay. Clear. So what audience tell DDBJ?
Japan.
Do you have any questions? No. On your side? Any questions? ನಾವು MCQ ತಗೋಬೋದಾಗದ್ರೆ MCQ ತಗೋಬೋದ ಎಸ್ ಆರ್ ನೋ ಒಂದು 5 ನಿಮಿಷ 10 ನಿಮಿಷ ವರ್ಕೌಟ್ ಮಾಡಿ ವಾಟ್ ಆರ್ ದ ನೋಟ್ಸ್ ಯು ಆರ್ ರಿಟನ್ ಅವ್ರದ್ದೆಲ್ಲ ಪಕ್ಕದಲ್ಲಿ ನೋಡಿ ವಿಲ್ ಟೇಕ್ MCQ to discuss among yourselves.
For the second session, that is research methodology.
Okay, so at home after this session, you can go home back and you can just access. See here, okay, so all databases option is there. So here, if you want the genome or gene information, so that you can select. So here, just to show you, I will just select nucleotide, so which is here. So you tell, so whichever the gene of your interest, so that nucleotide sequence will come. I think I will just type it cry1 AC, right? So once you give a search command, so you will get the information. See here at left part, so you just observe the cursor. So bacteria is there. So bacteria means that particular cry1 AC is found in bacteria. Okay. So which bacteria, students? Right? So it is there already, right? So that I will okay, so click here. So once you do that, so you will get the okay, the key locus. So where exactly it is present, what is the size of the DNA? So here 1070 base pairs. Okay, linear model. So always I used to talk about linear model. So here it is. Okay, so when it was published, so 22 December 2018. Okay, so next one, accession. This is the accession number. Version is 1. So what is the point 1? So if you edit it, okay, so after a few days with the new information, that version will come. Okay, so updated version. So organism is Bacillus thuringiensis. Reference is 1. Authors, so who are the authors? So their name it will come here. And what work they have done? Screening of cry genes from local Bacillus thuringiensis isolates, right? So journal they have not published this work. So they have just only published the sequence of the organism. Okay. So this is the source. So already you know that coding everything, what the program language. So that information it comes here. So which one? So this is the information and gene location. The gene name also it will come. And next one is origin. So this is the sequence. This is the sequence. Okay, sequence of the organism. So how it is organized? Linear alphabet model. So here one thing you can notice, in first row 60 bases are there. Okay, so next one is 61. It starts from 61. So here end is okay 1021. So this is just a bacteria sequence. If you just type BRCA1, it will take time to load. Just I will okay, if time is permitted I would have shown you, but it is very difficult now. So once you try it, okay, so it is very simple, students. See, now you want to run this, you want to get this primer. Okay, so you want to test on your plants. So what you need to do, you need to click on Okay, so pick primers. So it will give you the primers. So you know, okay, in PCR primers are very much important. So what are primers, students? Anyone? What are primers? What are primers? Primers is the short fragments of functional DNA. Okay, so they are functional in nature. So here I am in online class I will explain this. Okay, all this what it is. So if you just click on get primers, you will get both the forward as well as reverse primers. So which are used in the PCR. Okay, which are used in the PCR. See here. Okay, so the primer specificity cannot be determined as sequences of selected organism are not present. So none of them they are done. So in this context, so what you need to do, you have another software called as Primer 3. Okay, so or Primer 3 plus. So what you need to do, the same okay, the linear alphabet model sequence, you need to take that, you need to paste it here. Okay, so you are going to get the primers. So for that, what you need to do, don't copy this because it contains number. So what you need to do, you go back here, you click on the FASTA. Okay, so once you do that, so no numbers will come. So copy this whole thing, Control C, and here come here, Control V. Okay, so if you do that, so it will give all the again the commands. Okay, so what you need to do, you need to select some specific. Okay, so the specifications which you require by your related to your work, and click on the. Okay, so pick primers. So you are going to get the some of the functional primers, right? So that you can send it to the company. So here only one set of primer has been given here. It depends upon always the primer selection is depend upon the size of the genome of organism. Clear? And this primer, the ideal primer is having 16 to 20 base pairs. See, count the number of base pairs here. So which are present, students. Clear? Okay. So you practice this. After going home, YouTube lot of videos are there. So once if you practice, so you won't forget for your lifetime. Right? Any confusions here? Same set for EMBL also, DDBG also. Okay? Huh?
Yes, sir.
Okay, so research methodology, everything you need to know about research methodology. So I will just go a little faster. So whichever is important, so that I will concentrate. I think Rajesh sir already covered a couple of concepts in research methodology. Okay, so here, what is research? Anyone? Investigation of new thing or unknown facts. Okay, so research is nothing but investigation of new things or unknown facts which are already present. And also, for example, your friend has submitted some report. So whether it is correct or not, you can reinvestigate it. So that is also a research. Okay, so research again search. Okay, research again you search for what? For the truth. In scientific, so in the science, so you cannot publish the false results. So always it will be related to the truth. Okay, so it is like that. See, the first thing is, so one of the discovery of new facts, and second the verification of old ones. The object of every business organization, of course, the discovery of new facts, new relationship, and new laws governing the business phenomena. But constant verification of old concepts is also required for dynamic business development. So which is in context to the business. So I will just give you some of the inputs. So regarding what are these? See here, the different types of research are there. Quantitative research, qualitative, descriptive research, analytical and applied research, case studies, surveys, conclusive research, exploratory research, and fundamental research. See, all these are all different arenas of different formats of research. Okay, so if you are doing a survey, it is not that you are doing a bad job. So majority of MBA graduates, so they will do okay, research and surveys or case studies. So you can also do the case study. So if okay, some product will be there. If there, for example, one example I am giving. So if diabetes is controlling, so how many sample number you have taken? How many of they are found? So what is it? So what you need to do? So collect from which date they are taking. Okay, what is the blood sugar, glucose level? Clear? So that data you can take, you can publish the paper. Simple. No investment, nothing. Only thing is you need to have the frequent conversation with the patient. Okay, so descriptive research. So we will discuss all this. See, research is an organized and systematic way of finding answers to the questions. So if you are having any questions, okay, so the if you want to find a solution, so then the research will come into the picture. But how it need to done? So it need to be very much organized. However you want, you cannot do. Okay, so after going from college, so you know how you will behave at home. Okay, you are very much unorganized. You will throw stuff here and there, and the next day morning you will ask your mother. Okay, she need to get it for you. So that is how you are. Okay, so and systematic. You need to be very much systematic and disciplined, and you need to have some ethics. So we will discuss what are these ethics and other characteristics of research. Solution oriented. The problem of research must be clearly defined and stated. So what you want, that you need to clearly define. Okay. So next one is the motive of the researcher must be mentioned at the beginning of the research work. So you need to get some motivation from the who are already done this work or executed this work. If any failures are there, that you can do. Our life is not very big that okay. So we need to learn from the failures of others. Okay. So it is always better to learn from the failures of others. So what exactly happened? You need to get the survey. Okay. So you need to ask the people. So those who are involved in the particular research. The research should provide a solution to a business problem or any other thing. See the map my genome. Okay, know yourself. It is a Noida, Greater Noida, Delhi-based company. Okay, so what they will do? They will map your genome and they will tell. Okay, so the preventive measures of any forthcoming diseases. So that is what the service they will give, and their annual turnover is already crossed. Okay, so 200 crores. Okay, so the map my genome. It is an Indian company. So and 23andMe, one company is the US based company. So what they will do? So they will 23 means chromosome, set of chromosomes. Okay, so how nicely they have established a company, you see. Okay, so 23andMe. Okay, so 23 of my chromosomes and myself. So what are the the disabilities or forthcoming genetic variations and disease pattern, so that they are going to reveal you and they will give you a report. Okay, so and the periodical the checkups will be there. So this is again I wanted to just take this because necessity, right? So what people used to do, so they used to roam. So if you are visiting a new place for booking a room, so you used to hit every the lodges which are present. So now what is happening? So you easily through the app, so you can just look for the vacancies. So whichever the hotel is vacant, and also it will give you the best the price for your needs, right? Okay, the logical, it need to be logical. So illogical so things, so that is not coming into the picture of research. The interferences of generalizations does make me logical. For example, an illiterate people in a village. Okay, so example, an illiterate people live in the village live longer than the educated people in cities, leading to the conclusion that literacy is the cause of longevity. Okay, so illiteracy is cause of longevity, and this is the example of illogical research conclusion. You cannot compare conclusion. So like this is what illogical research. Okay, without any directions, right? So objective true picture of phenomenon without affecting by the observer's own opinion and termed as objective. So whenever you are start a research work, it need to be objective, it not to be a subjective. Subjective is if the four are sitting here, I have given one problem, so their solution is need to be same. It is not because since they are different people, so it is subjective their opinion. It should not change. Okay, it need to be objective here. Objectivity means knowing the reality. The criterion for objectivity is all researchers should arrive at the same conclusion after phenomena which they are pursuing on the research. So impartiality. A dishonest researcher may select data items of individuals to draw conclusion on his favor. So whatever the negative remarks which you get, see, any research it need not to be end with the positive note only. Okay, so it may end with the negative. So that you report. Okay, others they are not going to commit the same mistake. So that is one thing important you need to remember. So what many of them they will do? So they will manipulate. In case of healthcare, okay, never do that because if, based on your preliminary findings, so they are going to draw a conclusion. Okay. And brings bias into the research which affects the objective of the study. Accuracy, the research workers need to gain some expertise in the study is undertaking. See, now students are losing patience, okay, and they are not into their research. So what they will do? So they will just sit somewhere and they will write their results. So this is what it is happening. So that should not happen. So what they need to do? So they need to have some patience. And okay, so if the research demands repeatability, reproducibility, you have to do it. Okay, till you get satisfied. Okay, so till then, so you are not allowed to submit your research thesis. Accuracy of a conclusion is sensitive issue that may affect the whole decision making. So that is what I mentioned here. Okay, so however you want, you don't do it. So systematic. Okay, the well-defined steps. See, for example, tomorrow you are having lab. So whether the chemicals are present or not. Okay, simple. Whether that instructor is there or not. Okay, so whoever give the command, right? Whether you have got the procedure or not. Okay, so whether you are having all the adequate facilities are there or not. So whether the power fluctuations are there or not. So whether the instrumentation is present or not. So if you are working in a central lab, laboratory, so if you are having only 1 or 2 machineries, so you need to book in prior. Okay, so tomorrow from 10 o'clock to 1 o'clock, I am going to use this machine. If you fail to enter, okay, so if you fail to use that, please give a message to the whoever they have booked it later, so that you can manage it. So that is how you need to be systematic. So that you will learn when you hit a national laboratory. Till then, so however you want, you will have behave. Okay. Verifiability. Okay, the results of research are subjective and verifications. For building a sound basis of decision making, one verifies the research result by replicating the study. Okay, the verification is very much important, students. So that the verification will result to for better quality of the work. Next one is empirical or we call practical. A research is an practical process that involves data collection. The results are based on observed or experience or empirical evidence. The research rejects the assumptions. So there is no scope for assumptions. Your assumptions. If you throw this, so this will happen. No assumptions. You need to provide scientific evidence for this. Okay, and it accepts only what verified by the practical observations. See, you need to why you are maintaining observation book at your laboratory when you are doing practicals. Then quick, we don't have time. Why you will maintain observation book in your laboratory? Every day?
Note down the observation.
For note down the observation, right? That is only the purpose why you will maintain observation book. See, observation book is the reflection of your ethics and also how much you are committed to the work. See, in your research, so at the last you will go and publish this thesis. I will ask you where is your observation book or your raw data entry book. So if you fail to show that, I can directly say you are not performed the work experiment. You are understanding? So that is the importance of observation book. So for that, each one of you need to maintain the observation book. In our institute, so we have made it mandatory at the last examination. So you will be having 5 marks for record. So in our case, it is not that. 2.5 for observation, 2.5 for the record. Clear? Okay. 2.5 for the observation book, and we have excused many students who are not written the record. So observation book is must. Okay, mandatory. Okay, without observation book, how can an external examiner gives you the marks? Okay, so very important. Steps for preparing the research methodology. Okay. See, focus on your aims and objectives. Okay, so you must focus on your aim. See, listen. See, research need not to be a rocket science and breakthrough in something big. So you find the problems. Okay, for example, in BIT, another college, our competent college at down. Okay, so we are extreme opposite there. Okay, so rivalry college, whatever you think. So it is not that. So they have developed a wonderful product, sanitary napkins. They have produced from areca husk. Okay, so how many of you seen areca husk, students? ಅಡಿಕೆ ಸಿಪ್ಪೆ ಅಂತ ಕರಿತೀವಿ ಕನ್ನಡದಲ್ಲಿ ನೋಡಿಲ್ಲಲ್ವಾ ಸೊ ನೀವು ಅರೆಕಾಸ್ಟ್ ಅಂತ ಆಮೇಲೆ ಗೂಗಲ್ ಮಾಡಿ ನೋಡದೇ ಇರೋದು ಇಲ್ಲಾಂದ್ರೆ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಸರ್ ನಿಮಗೆ ಒಂದು ಸ್ವಲ್ಪ ಸ್ಯಾಂಪಲ್ಸ್ ಕೊರಿಯರ್ ಮಾಡ್ತೀನಿ ಅದಕ್ಕೆ ತೋರಿಸ್ತೀನಿ ಸೊ ಬೈ ಯೂಸಿಂಗ್ ದಟ್ ದೇ ಹ್ಯಾವ್ ಟೇಕನ್ ದಿ ಹೆಲ್ಪ್ ಆಫ್ ಟೆಕ್ಸ್ಟೈಲ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಅಂಡ್ ದೇ ಹ್ಯಾವ್ ಡೆವಲಪ್ಡ್ ದೇರ್ ಅಂಡ್ ನೌ ದೇ ಆರ್ ಇಂಟು ದಿ ಪೇಟೆಂಟಿಂಗ್ process and now the same institute is trying to do the sanitary napkins by banana pseudostem fibers. Okay, banana pseudostem fibers by in collaboration with the textile department. Okay, so our department we are trying to do the vetiver roots. So the lavanchada bear anta karitive, so which is having a lot of ayurvedic solutions. Okay, we are calling about now today we will talk about the reverse osmosis, that osmosis and this. So because of that scientists are now they are figuring out because of arthritis and knee replacement this RO water is main reason because of lack of minerals. So this is having in Kerala what they will do on the banks of this lake or water bodies. So they will prepare a bamboo mesh and on that they will grow these vetiver roots. So what this vetiver roots, it will absorb all the heavy metals, all the microorganisms. Okay, so and it will purify the water naturally without a single rupee investment. Okay, such a type of innovations you need to observe. Okay, so observe the problems which you are facing next to your home. Okay, garbage management, how best I can do. Simple. So such problems and such projects are gaining more importance in smart city. Okay, so projects, so which are all across the India. You are understanding, students? Clear? And agriculture based problems also they are gaining more fundings. Agriculture based. Okay, so because we all here too we cannot eat mouse and keyboards, right? So you need rice and you need dal, right? You need other raw materials, ingredients, wheat you require. So how you are going to help the farmers? Okay, with all your innovations, right? Focus on your aim and objectives. Identify research method that suits your research problem. So now you have shortlisted one problem. How you are going to do it? Next question. Okay, so how you are going to do that? So for that, okay, so what you need to do? So you need to read lot of literatures and you need to come for a conclusion. So I will do it. So don't do however you want. Please ask the expertise opinion. Okay, so rational the selecting particular research method. Okay, the same continuation to this. Consider your audience and think how you will collect the data from the study. Okay, whether the data, okay, so of your interest, how you are going to collect. So for example, if it is based on clinical trials, okay, whether you are taken the permission, okay, so from the clearance certificate from animal court or whatever the required premises, or from plant, whether you have taken the permission from the institute, okay, or for the usage of the material. Okay, so that you need to think. Analyze the different types of data collection methods available and identify the data collection method is whether it is feasible or not. See, at the some point of your work, you need to stop and analyze whatever I am doing, whether it is really helpful for the people, common people. So that you need to question yourself. Okay, so that small ethics you need to have. Okay, and next one is analyze the different data analysis techniques that suits to analyze the collected data. See, analysis is very important. ANOVA, you know, stats you odidira, biostatistics. Okay, so if you study biostatistics, there some tools are there. ANOVA. Okay, so the two objects variability, so that you can study by doing ANOVA. And the graphical representation. Okay, after going home, you please Google it and check the applications of ANOVA, right? And the next one is how to plot a graph. Okay, so you cannot present the raw data as it is. You need to take the mean of the 4 samples, and that mean you need to put it into the average, and that average data you need to plot a graph. So then it will be readable format. Okay, so that is what it will tell you, analysis of the collection data. So research process, problem formulation, literature survey, development of hypothesis, research design, choice of sample design, data collection, analysis and interpretation of data, hypothesis testing, interpretation of results, and at last report writing. Okay, so these are the process. Okay, so they important in the research process. Okay, so problem formulation already I said, literature survey clear, development of hypothesis. If this is happening, what will happen to that? This is what the assumptions. Okay, if the metro pillar is this much cubic meter of cement, so whether it will sustain the next. So such a type of hypothesis and questions it will make aware of these engineers, right? So what they will do, so they will give for betterment or better compatibility, or they will add fast setting gypsum in it. Okay, so that is how they will change themselves. See the significance of research in the psychology. So it is different. Okay, the number of people with some medicine. So how they will behave, right? How they will behave in different contexts. The people, those who are addicted to drug, rehabilitation centers will be there. Okay, the drug addicts. So how you are going to change the psychology. Okay, so that significance of research you need to tell. So in sociology, socio-economic manner, how that research is going to helpful for the society. And next one is economics, how it is going to helpful for the development of economy or whatever. And in geography already in bioinformatics I have covered it. In education and very important in medical science. So what is the significance of your formulated idea in the medical research? So is there any phytochemicals? What are phytochemicals? Plant-based compounds. Okay, so they are called as phytocompounds or phytochemicals. So so for curing of many diseases, for anxiety, so some herbs is there, right? So how you are going to conclude it? So based on many results and trials. So without clinical trials, you are not having any right to release into the market. Okay, so you need to have a FDA, Food and Drug Administration Authority clearance. Now earlier it was there. Now Indian regulatory survey, I don't forget. I just forget that agency name. So you need to get the clearance certificate them, and you can do it. Okay. And business management and in government functioning also, the significance of research will impact. See, for example, you have found out some solution for the agricultural problem. Okay, so that solution, for example, related to the irrigation, so that you can give the technology to the farmers. You are understanding? So that the farmers also, they are very well aware, and government will transfer the technology. Okay, the government will transfer the technology. It may be solar irrigation because of power fluctuations. Farmers, when they will go for irrigation, midnight. When the whole world is sleeping, they will go to the field by 11 o'clock, and they will come to home by morning 5 or 6. So this is the condition. Okay, so on what our life is depending, so they are suffering there. Okay, so to supply food grains and staple food for the mankind. So this is the situation. How best you can see, you have a solution, sir. Solar pump sets are there. Okay, they are very costly. They are not affordable. Is there any solution? See, in our premises in RV College. So I think all of you heard about RV College. So he is a mechanical engineer. He has developed or he has outsourced drone components, and he has developed a drone. And what does that drone will do? So he is talking about the drone Pratap. So not a problem. Okay, so see, he established a company called Greeny Fly. He established a company called Greeny Fly. So you know what is his? He is still 8th semester student, engineering student. Okay, he is a 8th semester engineering student. He will come out with the solution. Since he belongs to that Shirsi region, so there a lot of paddy is being grown and no manpower. As I mentioned, 140 crore population, no manpower for spraying the pesticides and chemicals. So he has adapted 10 liters of tank. Okay, he has attached, and if you fill these pesticides, it will go and geotagging everything he has made, and boundary will fix. It won't go to your field and it will spray. It will go to the whom field it has to do. There only it will go. You know the cost of for 1 hour. So it will cost 1 acre. It will cost 600 rupees only. So for labors, you just imagine for labors you need to give whatever that gutla whatever he chews, alwa, and you need to give meal and drinks also you need to give at villages. In fact, the harsh reality I am saying. So now with this advancement and if he inhales the pesticide residues, he will die. If you drink and go to the field and spray the chemical pesticides and fertilizers, he will die because of the adverse effects. Clear? So such a type of innovative things are happening in and around us. So only thing is you need to look. You please have all of you the practice of reading newspapers, whichever, so that you will be aware of what is happening. Otherwise, so you are not connected to the environment. You are connected to the only the social status of your friends. It is not going to helpful for your life. Okay, please connect to the real and the groundbreaking technologies which are all coming day to day life. Okay, and the government functioning. The problems encountered by these researchers, lack of skilled researchers, that is there because you are not having patience. Okay, so if any failures are there, you will cry. Correct? So you need to have the tendency, repeat it till you achieve. You have to do it. Okay, so Thomas Alva Edison bulb story. I am not going to repeat. You just Google it later. Okay. So again, it will become 15 minutes time. Okay, so wonderful story is there behind bulb how he 100 times he failed, but at last he succeeded. Okay, now whenever you talk about light and bulb, so you will remember his name. So like that. The lack of code of conduct. So this is very important. So where discipline is not there, there Lakshmi also won't come. Saraswati also won't come. Right? Okay, the insufficient interaction. No interaction. Okay, no interaction. So after the class, okay, you will carry with the doubts and you will end up with the half-boiled knowledge of Google. Right? Half-boiled truths you are going to. So overlapping research studies. Already I mentioned the same thing is carried on here and there. It is not going to useful for either of any. So lack of confidence. Okay, so no confidence. Okay, no confidence. See, in so in our college, so whoever they got 99.5%, they are not got displacement. Okay, who is having 75? So you got because of confidence. So they don't know how to interact. Okay, the communication skills we call. So that is why I tell each and every student, you interact. How you interact? The more you interact, you will get job faster rate. Okay, the faster rate into 2. How you skip your videos into 2x zoom. Okay, so likewise. Okay, right? You are understanding. You interact with the faculty. You interact with the society. So whoever sitting next to your bus, okay, the passenger interact with them. Okay, so because maybe you don't know which scientist he is. See, opportunity is when you interact with the people, it will come. You never know. You never know who he is. Okay, until please stop. Watching the mobiles. Okay, so when you are doing journey. Okay, so interact with the people who are sitting next to you. So I found a scientist. So when we are traveling to Chennai, so belongs to the same institute, and he helped us in all the manner, including from arranging the guest house also. So we don't know who he is. So we are heading to the MS Swaminathan Research Foundation. So you know MS Swaminathan. Who is MS Swaminathan? He is the father of Green Revolution in India. So we are there to analyze for the rice paddy. So already in introduction they said I did my PhD on micronutrients in rice, zinc and iron. Okay, so for that analysis, only two institutes they are having, Acharya Rangarao Agriculture University at Hyderabad, and one more is MS Swaminathan Research Foundation. So every 6 months we used to go there. So there, for the first time I met the, okay, he is the next, co-passenger next to me. So we don't know who he is, right? So if I didn't talk to him, I would have struggled a lot to reach the destination. Definitely I would have gone there, but lot of effort. So what he has done, he has already the institute car, so he took me with this car. So likewise, the contacts will also grow. Okay, so never restrict yourself. So no one is asking for your property. Okay, so who are sitting next to you, so they are not going to ask you for the property. Okay, so bond paper itkondu bandirtara, sign ಮಾಡಿ ಅಂತ no. Interact with the people. Okay, the good people, those who are sitting next to you. And this is common, obvious lack of funds and facilities and lack of literature. So these are all again important components. See, the word saying goes, necessity is mother of invention. Okay. So when the necessity is there, definitely you are going to invent, right? So I will just skip on this. Okay, the features of good research is freedom from bias, freedom from confounding. Okay, the freedom from exogenous variables, statistical precision and testing hypothesis. Okay, within resources, optimality and objectivity and flexibility. Optimality I will stress more here. See, you need to be very much optimistic. So in our class, so if one student is sitting, we will teach him because he has come here, okay, a long way, and he want to sit in the class. So why we need to look for rest of not present in the classroom? So we need to be optimistic. At least one student is sitting in the classroom. So that is how you are going to grow. So already one story is there. So a professor shows a empty glass. So what it is full of? Empty gas? It's empty. So you will say, but a optimist will say the glass is full of air. Okay, so next half of the glass is filled with water. So what is present? Half glass is filled, the negative. Okay, activist will say, but optimist will say half of the glass is filled with water and half of the glass is filled with air. So for a successful life, so you need to be optimistic and be optimistic. See, because in this generation, so in a small hurdles in life, they will commit suicide, right? So this is the harsh reality. So they are introvert in their life. See, education is not only for finding something and doing. No, be a good daughter and son for your family. So that is also a great contribution you are giving to the society. Remember. So here I don't know your background, students, how rich you are, how your parents are managed to make you sit in this classroom, and you need to know the purpose why you are sitting in the classroom. Okay, so you need to know what is the purpose of sitting in this classroom. Okay, I know my purpose. I am here to teach bioinformatics as well as research methodology for all of you who are sitting here. So you know what is your purpose. Okay, you know your goal. So that you need to highlight, and whenever you are want to commit any mistakes, you remember your parents in front of you. Okay, so any mistakes if you are about to conduct, if you are about to do, so please remember your parents. So you will be a better person tomorrow. Remember, students. Thank you. If anyone interested for internship, you can go there. Okay, for food industry. Frozen foods, they will make jackfruits, because in malnutrition the jackfruits will get vanished. So what they will do? They will frozen it and they will send it to the other countries. So this is what you will do. Okay, the frozen food, very big market in other countries, right? See, after your completion, listen. After your completion of your degree, so you can do bioinformatics is there. So I don't think bioinformatics is present in Bangalore University. Please check it. Vellore Institute of Technology, VIT. Okay, very good institute. There it is there. Parle University, Vadodara, Gujarat, it is there. Okay, so one of our senior is head there. Okay, if you really want to pursue there, you can do it. And of course, by MSc Biotechnology is there. So if you are coming from zoology background, CZBT also there, CBBT also there. So you can do masters in both botany as well as zoology also. Because the NEET aspirants, their lack of good resource persons. Okay, so and it is not that the tutors are not getting enough money. So one of my friend is a NEET coacher, so he will get okay, so 10 to 15 lakh package in this Parishrama as well as one is there in the base academy, base and Akash. Okay, so it is not that tutorials you are not fetching money. One session they will learn 1 hour, they will learn 3 to 4,000 rupees. Students, only 1 hour of teaching. Imagine. So how big the field is. But the problem is admissions for MSc Zoology as well as botany is drastically okay, come down. So I don't know. So that field in coming days it is going to face a lot of okay, vacuum in the market. So only gap is. So in that area gap is there. Clear? And after that in Bangalore City also there. Okay, so if you want to like do some research in agriculture, there also you can approach. Soon after completion of your okay, degree, you can go there. But there they will only accept the MSc graduates in agriculture university. In NCBS also, you visit NCBS if you really want to. If you really want to do okay, so PhD, integrated PhD they are offering. So they will take a entrance exam. I think June, July they will release the notification. And but unfortunately, only North Indian students they are getting seats there. So because you are not aware. Okay, so what is happening in the JNCR also, Jawaharlal Nehru Research Center at okay, Airport Road, Ballari Road, you know, Airport Road. There also Patrayanapura. So there also one institute is there. There also you can join. And medical, okay, central medical as well as is medicinal plants institute is there. There also you are having ample of opportunities. For I am just talking about the okay, higher studies. So what and all you can do. Integrated PhDs are there. So everywhere in IISc also there. Okay, so that you please concentrate and join. Okay, so please make note that in which area you are interested. After completion of your MSc, Nimans also they are offering PhD. Okay, so PhDs for aspirants. Otherwise, if you want to do a job at it is better to earn from the young age. Okay, so that once you become expertise, so definitely you are going to get the very good salary. Okay, at the end. So don't be very ambitious and soon after completion I need to get 5 digits. No, it is not going to happen until unless you are skillful. Until unless you are okay, different from your friends. See, now all of you complete the BSc, right? How I can distinguish from you and you? Tell me. Skills. Okay, so you need what you need to do? You need to stand apart from the crowd. So then you are going to achieve something. It is not the end, dear students. Prepare for KS, IAS, competitive exams, banking sectors are there. It is not that. If you are not interested, you please look into your family background and financial okay, so circumstances. So accordingly you plan. Okay, so accordingly you plan. So if you want to clear, okay, so if you want to do PhD, so definitely please don't go without funding. Okay, without fellowship, don't join. Okay, so without fellowship, don't join. So for that, what you need? CSIR net or net exam you clear. So you are going to get 36,000 rupees stipend. Okay, clear? Or any agencies, any institute you want to join, so please join whether funding is there or not. Otherwise, you are going to suffer in the future. Without funding, please don't join PhD. Okay, for that, what you need to do? That you practice from today. Start from today. So by completion of your 2 years of MSc, so definitely you are going to clear the CSIR net. Okay, clear? And a lot of vacancies are there. Okay, in the government sector also, in private sector also. Look for KMF. Recently they have called for the graduates. Okay, KMF, Karnataka Milk Federation. So many are there, many okay, regions are there. So there also you will get. In all the and forensic department, main Karnataka state, Okay, Karnataka State Police Department, KSRP, something is there. Okay, so there also opportunities are there. Okay, government opportunities. So after completion of your BSc graduation. So anything if you want to know, please ask Rajiv sir or myself. So anytime I'm here to clarify your doubts. Thank you.
Thank you, sir. ಮಾತಾಡೇನಪ್ಪ
Now, I request Dr. Pratibha K.S. Ma'am, Head Department of Biotechnology, to present a token of gratitude bouquet and also the journals for the sir, which he have been awarded from the Department of Biotechnology for the best oral presented award in which was conducted by the Department of Biotechnology in the year October 2020. Best Oral Paper Presenter Award for presenting entitled Implementation of Artificial Intelligence for Sustainable Agriculture.
Today we went through two sessions, right? One is introduction to bioinformatics and second session you have heard today, you were good listener, you were attentive, I can make out that one. Whatever information you have written, you please go through that one. If you have not received or if you have not understood thoroughly, the next again online session sir is going to come. That time also you can interact with sir. Is this clear? Day one, I do agree that it's quite difficult to interact and ask questions like that. Next, when we join online next coming Monday, I'll just suggest sir that when is this and when we are available, sir will change the Shada Sabha and here only online session will be going on. Are you ready to listen second session again?
Yes,
We know third session. Clear? That time if you have any kind of questions or any queries, any doubt, that time also you can clarify. Or you can also write to email ID. Instead of going to the contact number, write in email ID. If you have any kind of queries, you can discuss over there. Can we have a big round of applause for Under 5 minutes, please go through whatever you have written. 5 minutes, you plan that one. 3 by 1.
Okay, so please leave your info outside.
ಪಾಸ್ ಮಾಡಿದ್ದಾರೆ
So otherwise
ಇವಾಗ
they are not going to entertain. Okay, so they are just there to check your patients. So if you want to get into the any reputed institution, at first glance they will reject you, but don't quit. Okay? So that is what.